Okay, we will start today. We stop at the MD receptors, and now we're going to talk about the AMPA receptors. AMPA receptors is also neurotrophic receptors. This is also widespread in the central nervous system. Now, actually, it is parallel distributions to the MD receptors. Now, you can see that actually is involved in the so-called fast component of the excitatory postsynaptic potential, which means that when the glutaminergic transmission started, the response of AMPA will be the faster one, okay? And until that, the depolarization enough, then that will unblock the magnesium in the MDA receptors, then the MDA receptors then will be function and transmissions of the signal into the neurons, okay? So that's the reason why the AMPA is also very important in the case of the transmissions, glutaminergic transmissions. Now, kainate, the other one, is also a neurotrophic receptors. Now, is more concentrated in a more specific, a few specific areas in the CNS that complementary to the MDA and AMPA distributions, particularly in the striatum, lucidum, and also the lucidum region of the hippocampus. Sometimes it's difficult to distinguish with the AMPA because of some of the crosstalk of those agonists. But if that is present at the absence of the AMPA and the MD receptors, particularly, for example, in the dorsal root C fibers, the those are sensory neurons fibers, then actually that is more easy to detect or specify the study. Now, so we talk about the glutaminergic system, how the glutamate are being synthesized by the linked to the Krebs cycle and those enzymes. Then we will talk about drugs that acting on the glutamate receptors. There are not a lot of very useful one, but actually this system actually have the potential therapeutic agents because that, for example, treating the epilepsy and also neurodegenerative disorders as well. So as we mentioned about, some actually there are not small amount of people suffering from epilepsy. So actually there are some percentage of those people suffer from epilepsy, and some of those, if they are too serious, and then they may need to do surgery in some of the very, very serious cases, okay? So there are main types of excitatory amino acid antagonists and select for their main receptor subtype, receptor types, but not for very specific subtypes. A lot of those are unable to penetrate the blood-brain barrier. So those are not very useful because that if they cannot cross the blood-brain barrier, then they will not be able to add on the central nervous system. Excuse me. Okay. Now, some of those that has been studied quite a bit is the MDA receptors, because in the central nervous system, if that is, for example, during the causes of a stroke or during the neurodegenerative process, it's believed that is contributed by the MDA. So when we talk about excitotoxicity, then you will know more, because one of the reasons behind is that when MDA receptor is open, then calcium can rush in as well. So too much calcium in the neurons actually can cause the cell death actually. So as now you know that the MD receptor got the glycine binding site as well, that one actually can be used. If you can block that site, then it can serve as the antagonist as well. So So that's the reason the quinonic exit actually has been studied and see whether they can add on this way, and also other compound as well. Okay. Now the actually the one that has been used are the channel blockers. Which different substance actually act on it. For example, ketamine and phencyclidine actually, and also supine, MK801, and the memantine. Those are actually one that blocking the channel, especially the one that has been tried and prescribed. This is the memantine actually has been used in the treatment, or not quite treatment, actually prescribed for people with Alzheimer's disease. So those are the channel blockers. Now those channel blockers can pass through, because they are lipid soluble, they can pass through the blood-brain barrier. Okay. And ketamine, you heard about it quite a lot. So that one is also, can be used as the channel blockers. Okay. There are also competitive NMDA receptor antagonists, for example, AP5, and some other experimental compounds. So that has been explored. So that actually an NMDA receptor has been blockers, antagonists has been actively developed, but still so far the usage is very limited. Not many drugs actually successful and very useful acting on that. Okay. So then we will go to the another neurotransmitter, the GABA, gamma aminobutyric acid, GABA. Or now GABA neurotransmission, that's the inhibitory neurotransmitter. It's very important as well, because if this is add-on, then that can suppress the transmissions. And one of the very important drugs, actually so-called the anxiolytic drugs, is acting on this GABA neurotransmitters. Okay. Now the GABA occurs in the brain tissue, but not in other mammalian tissues, except in trace amount, mostly in the brain tissue. So around 20 to 30% the synapses in the brain use GABA. Particularly in the nigrostriatal system, and that also have implications on its effect. Now the GABA coexists with the neurons with the 5-HT acetylcholine and also peptide neurotransmitters. So it is usually released by mainly short interneurons, so serve as inhibitions. But there are also some of the long GABAergic tracts running from the cerebellum to the striatum. Okay. So actually, all neurons are sensitive to this GABA inhibitory effect. So its function is ubiquitous in the brain. So actually, so that's the reason why another compound, ethanol, actually alcohol, is also acting on this as well. So the synthesis of the GABA, GABA is formed by the metabolic pathway, so-called the GABA shunt. The glucose is the a precursor going through the glutamine and also glutamate. Okay, so from the glutamate, by the actions of the glutamic acid decarboxylase, so it's actually the carboxylations of the glutamate, then they will form the GABA. Okay. Then how that is actually inactivated? So it's by the alpha oxoglutaric transaminase, GABA transaminase. GABA transaminase. Is the catabolic enzyme that transferred amino group. So you see that from the GABA to the alpha ketoglutarate, then will become glutamate. Then that can become the precursor for the GABA again. So you can see that actually it's here glutamate, remove the carboxyl group, then it becomes the GABA. Okay, and GABA remove the amino group, then that will become the succinate semi-aldehyde. Now this can be going to the succinate exit and then go back to the Krebs cycle. Okay, you should still remember those. So you can see that actually it's very similar to the glutamate synthesis. So in the neurotransmitter and the transporter in the glutaminergic synapse, that's very important. This structure of the transporter actually with a 12 transmembrane subunits, both the N and C terminal in the cytosol. The loop actually contains M3 and M4 actually are being glycoprotein. Okay. So it depends on, again, the sodium and also the chloride concentration at the ion for the transport, you know. So here is what happens in the synapse. So in the neurons, is glutamine actually will be with the glutaminase changed to the glutamate, and then the GABA exit the decarboxylase actually be made to the GABA, and then that is actively stored into the vesicle, and then it's released. After release, it will the reuptake transporter. You can see that both either you can go back to the neuron that can be reused again, or a lot of those are actually taken care by the astrocyte. Okay, so that we move it really fast. So you can see that, you know, from the terminal, it can be utilized. Now in the glial cell, then that will be metabolized to succinyl semi-aldehyde. So you can see that the GABA with the transaminase that will become the succinyl semi-aldehyde that can go back to the Krebs cycle. Okay, and the amino group will be using the alpha ketoglutarate that will change it into the glutamate, and the glutamate will go back to glutamine, and then the glutamine can pass it back to the neurons, and then you can get the cycle again. Okay. So that's the reason why that, you know, the astrocyte actually is helping quite a bit in both the glutaminergic and also the GABAergic neuron transmissions. So if you still remember, we talk about the astrocyte are helping the transmission, actually very important. So how about the receptors, the GABA receptors? So there are two distinct types of receptors, the ligand-gated channels, the GABA-A receptors, and the G protein coupled receptors, that's the GABA-B. There's a less study that actually most studies the GABA-A receptors because of the pharmacological importance of this group of receptors. Okay, the GABA-C actually is similar to the ligand-gated channel. Okay. So again, this is you can see that there are alpha 1 to alpha 6, beta 1 to beta 4, gamma 1 to gamma 4, and there are also the other minor one. You can see that the identity actually across and down. So you can have all these combinations so that to make the channels more sensitive or less sensitive depends on the places, and also the neurons respond. So that's the reason why that is always like this. There are different subunits, so that you can have different subunits, you can have modulated different, so depending on the neurons. So the GABA-A actually belongs to the same structural class as aconium receptors. They are pentamers, mostly composed of the different subunits, alpha, beta, and gamma. Okay, so each of those, they have can exist 3 to 6 molecular subtypes, as you can see that over here, you know, different subtypes. So they have the putative membrane spreading helix. You see that over here. And there are some glycosylation site. There are serine phosphorylation site as well. They can modulate the activity of the receptors. So that can actually desensitize. So you can see that a lot of ways to control the receptors. So that's the reason why that in the nervous system, the action is so versatile that you can make it more active. You can make it less active by those different methods, for example, phosphorylation or endocytosis, and modulate the amount at the surface and also modify outside so that they can be less sensitive or more sensitive. Is it okay? So the GABA A receptors is the postsynaptically immediate fast postsynaptic inhibition. So yes.
You were saying it's a pentamer, right?
Yeah.
GABA A receptor is a pentamer. But then in the previous slide, you depict it as two tetramers.
No. It's just illustration of the membrane column.
So one of each of the four membrane domains on one subunit.
Yes. Okay. So this is just illustrate, for example, alpha and beta subunit. Okay. So so it open up and then lets the coral ions to go into the neurons, so that that cause the hyperpolarize the cell. So this causing hyperpolarization. So this is called the inhibitory postsynaptic potential. Okay, so it's going down. So that's how the integrations. So if you still remember, we talk about the neurons receive the desensitizations here means that internalization of the receptor, page 34. Oh, sorry. No, it's less sensitive. Okay. So that caused the hyperpolarizations, as I mentioned before, that actually the neurons will receive all the inputs from different connections, the synapse. Some of those will be excitatory, some of them are inhibitory. So once that is integrated all together, if the membrane potential is enough, channeled to the axon hillock, if it is enough, then that will initiate an action potential. Oh, okay. So this is something simple, but when we talk about the other modulator or neurotransmitters, then that can also integrate the information as well, depending on how they modulate. So that's the reason why that some of those neurons, some of those neurotransmitters can change the perceptions of the human's mind. Okay, when we talk about when you do the your projects, then that will be the time that you can look at some of those. So speaking of the project, I just received one more email regarding the grouping. So by the beginning of next week, then I will arrange the groups for you, and then we will talk about the group topics and also the assessment method. Okay. Okay. And we have finished the midterm, and the assignment will be submission deadline is coming. Some of you have already submitted the assignment. If you haven't, then please remember to submit. And in November, I will release another assignment as well. Okay. So we are halfway through. A little bit strange. We have just a few students here. Some students, most of you are online. So anyway, every courses are like this, I guess. Okay. So drugs that acting on GABA receptors, GABA A receptors. So you can see that there are agonists and antagonists. Barbiturate, this is one that actually used in US. This group of drugs actually used in US for execution of those death penalty, actually, as barbiturate. And ethanol depressants also believed to act on the channel as well. Now, this group actually benzodiazepines. Actually, this one is helping the agonist. Benzodiazepine actually is a very famous drugs that actually a lot of people abuse it because it's anxiolytic. Okay, I'll talk about what is anxiolytic. There are steroids that can buy as well. There are also other drugs. Okay, so there are agonists or antagonists. Okay. So when we look at this group of GABA A drugs, the benzodiazepine or diazepine, benzodiazepines and or diazepine or Valium is the one that actually abused by a lot of people because it's anxiolytic. So we reduce the anxiety of the people. For example, the famous one, for example, Michael Jackson's, he was taking a lot of those benzodiazepine actually, or his doctor was prescribing him about that particular before he died actually, but he was not because of these drugs that actually passed away because of another drugs. Okay, barbiturate sometimes used that as anesthetics. Okay, now the anxiolytic also has a potential. This group of drugs actually potentially being abused because this particular in 1980s when people are not quite sure how the effect will come, because in the Western countries at that time a lot of housewives are being prescribed of this because the housewives they have a lot of things to take care of the children and housework and so on. So doctors will become anxious, being anxious. Then that is something that for example, when you are going for the exam or midterm, you're anxious about whether you can know how to do it or get a good grade. Then you'll become anxious, and if you take this kind of drugs, then actually you get anxiolytic and help to make you more peaceful. However, because of the nervous system, it's usually you have to pay for it, because that if you act on it all the time, then the receptor may be desensitized or downregulated. Then you need more before it has the effect. So that's the reason. Okay. Okay. GABA B, actually, those are not very useful. Those are not actually being used very much in the clinical treatment or pharmacologically. Okay. Now, that's what we talk about. The target, the GABA A receptor target several important drugs. Normally the benzodiazepines, barbiturates, as I talk about, and neurosteroids. But benzodiazepine, Valium, actually regulate the GABA A receptors. So you can see that when you help to activate the GABA receptors, the overall sense of the person will go down. So it's more calm and not as anxious as before, and also related to, because a lot of those benzodiazepine actually is within the striatal circuits, that also related to the reward center or addiction center as well. Okay. So again, so don't worry too much about those drugs names. Okay. GABA B receptors actually is located pre and post synaptically. They are coded in 1997, and then it's function as a dimer. It actually have inhibiting the voltage gauge calcium channels, so that by opening the potassium channel. So, but GABA B actually receptor has not been studied in detail, particularly because this is G protein coupled receptors, so that it's a little bit more difficult to study. Whenever the result, so it's a fast acting response, changing the membrane potential, those will be receptors better studied. There are some experimental compounds produced antagonists. Sarcophan actually seems to be able to penetrate the blood-brain barrier, but the effect is not as expected because it's more like anti-epileptic actions. So that's the reason why that this group of drugs is actually not being used or not having big importance. Okay. So the last amino acid neurotransmitter, the glycine, mainly in the gray matter of the spinal cord. This one also has not been studied very much. We know just that it's in the gray matter in the spinal cord using as a neurotransmitter. It seems to be inhibitory hyperpolarizations. Okay. Sorry. So it's not well studied. Now the receptor actually is similar to GABA A receptors. It's multimeric, ligand-gated chloride ion channels. So it's, also have the inhibitory effect. You can see that no therapeutic drugs has been add-on study for these transmissions. Okay. So we finished the amino acid sections. Any questions? Face-to-face student and also the online student.
Hello, Professor.
Yes.
I actually have questions regarding page 30.
Uh-huh.
And you mentioned that the astrocyte would change the GABA into glutamate and then into glutamate and then into GABA again, this cycled. And why don't they just make a storage of the GABA? Why do they not need to break it down and then make it into glutamate and then make it into GABA again? It's kind of energy consuming. It's not very efficient to do this. Why they design this kind of mechanism?
That is a very good point. Actually, the astrocyte cannot. So if you think about how you can transport it between the astrocyte and the neuron, you can see that they change it to glutamine so that it will not affect the outside concentration. So if you just pass the GABA outside, so how can you pass? So you cannot do exocytosis and then endocytosis. Okay, but if you change it into something that will not affect the transmissions, then that will be a much better approach. The same as the glutamate. You still remember the glutamate also do the similar things, uptake of the glutamate and then convert it into the glutamine. Then glutamine can pass out and it will not, affect the transmissions. Because here is you want to remove it as soon as possible and then convert to something that actually will not affect the neurotransmission outside. And then at the same time, you can pass it through to the neuron so that the neuron can reuse it again. So that's the reason why the astrocytes are actually doing the supporting role that in the transmission. Is it okay?
Yes, thank you.
Okay, good. So you will see more and more example of, the removal or how the metabolism of those neurotransmitters. You see that we have seen that two major methods. One is the enzyme metabolize it outside, so that's the acetylcholine. Okay. The other one is that actually the uptake that is actually in the both the GABA and the glutamate. Okay. So any more questions? Does astrocyte also obey the Dell's principle? That's not Dell's principle. Means Dell's principle is about the neurons are using the transmitter, or the neurons, the same neurons will use the that neurotransmitter. So it's different for the astrocyte. Astrocyte is not, how to say, the astrocyte is not transmitting signals to the or forming synapse to the other cell. It will have signal given to the neurons. For example, it will secrete, for example, growth factor to support the neurons, but they are not the principal information transmission or transmission of the signal. Okay. Oh no, this is so depending on types of the astrocyte, if they're around, then they would. So it is not taking care of one neurons or whatever. So in the surrounding, it will uptake. If they have the transporter, then they will uptake. They can do both. Okay. So it's not like one neurons, one astrocyte. So because you can see that the neurons actually have long projections and project to different areas. So but astrocyte is actually more localized. So it's not like one-to-one. Okay. But interesting, yeah, the way that you think about it. So any questions? Any more? If not, then we start the next neurotransmitters. Okay, category A mean. Okay, we have. No, they are not. The astrocyte will not just specify in the metabolism of the one neurotransmitters. So it helps because you can think about that. In particular location, there will be not just one type of neurotransmitter being released. There might be several. Okay. Okay. So category A mean actually is important category of neurotransmitters, both in the peripheral and the central nervous system. Now, obviously, the functions of it in the peripheral will be quite different from the central nervous system. And let's look at those. So this group include the catecholamine, dopamine, and noradrenaline. They are essential neurotransmitter in the brain. They are non-adrenergic neurons. A lot of important targets for drug, both in the central nervous system and in the peripheral. Now, peripheral will be including the adrenergic in the sympathetic system. Okay. Now, however, in the central nervous system, that also have this class of neurotransmitter also have important functions in which I always like how you can they are in more organized pathway in controlling some of the for example, happiness or rewarding. So, for example, dopamine actually is related to the rewarding system. Okay, so just like how, so you say, for example, how eating can be very happy or not so happy, Now, some of the things that actually make us to do is because of this kind of setting the tone of you. So, as I mentioned in the before, the addiction is actually is making us to survive. Okay, if we don't eat, we're going to die. So if you eat and you feel happy, there's a feedback for you to make you to do the same things again. Okay, so there are circuits that actually change the perceptions or your interaction with the environment and help you to survive in the environment, well, or response to the surroundings. Okay, for example, we are facing a lot of challenges nowadays. For example, we cannot go out to travel. Basically, we cannot go anywhere unless that you have to quarantine in the other countries. If you are not the resident of other countries, you cannot even go there. Like a lot of people, a lot of people like to go to Japan. So nowadays you cannot even go, or even we cannot go out that often, or going out to eat or with friends, or watch movie. So how can we, cope with those, even just the same? So those are the time that actually we need to survive and then adjust, Now, this curve of the neurotransmitter somehow is setting the tone. So compared to the glutaminergic and then GABAergic, those are the working neurotransmitters to initiate or control your movement or behavior. But at the same time, there are this class of neurotransmitter, particularly catecholamine or another the other one, the serotonin as well. Those are the ones that setting a different tone, and those are also actually more difficult to study. Now, for example, in the noradrenergic pathway, there are different pathways. For example, mainly this group of neurons in a small pocket, and then they will innervate it, send the innervations out to the whole different brain. Okay, for example, the locus ceruleus actually is here. Also the brain reticular formations, and also the nucleus of the tractus solitarius. Actually, those are the small group of neurons, and then they will send extensive output to different part of the brain. Okay, some of those will go to the amygdala, some of those will go to the hypothalamus. Okay, this will go down as well. So you can see that there extensive. Now, this one is actually different from the sympathetic pathway, okay? Because those outside, if you still remember in the sympathetic pathway, from the preganglionic, so that's from the spinal cord to the postganglionic neurons, the ganglia. That one is acetylcholine, and then outside the ganglia, actually those one use the noradrenaline as well. Same as in the adrenal gland, that you also use that as well. Those are different. So okay. So this one is in the central nervous system. Now the other one, dopaminergic pathways. There are also a group of neurons in different areas. So there is a substantial nigra, ventral tegmentum, in the olfactory tubercle, neostriatum, nucleus accumbens. Those are the projected areas. So there's in the substantial nigra in the midbrain actually, and ventral tegmentum. Now it will send those pathway into frontal cortex and also in the striatum. Okay. Those actually, particularly the frontal cortex, nucleus accumbens here, and the striatum, nucleus accumbens actually are related to the addictions. Okay, so that's the reason why that is related to the addictions, this pathway. And if people, so some people are more prone to addiction, so it might be because of their differences in the connection pathway. Okay, so the nigrostriatal pathway, the mesolimbic pathway, and also the hypothalamus pituitary pathway as well. So you can see that going to the hypothalamus actually is the hormonal part. And then, you know, now first special, this pathway, apart from the addiction, there's also control movement as well. This is the pathway that actually control movement. So that's the reason why in the disease, if we have time to talk about the Parkinson's disease, is the disruption of this pathway. Okay. So that's the reason why that some of the example, those people with those Parkinson's, they might have other psychiatric disruption as well. Like, you know, some of those are more easy to become addiction as well. Okay. So another category is the biogenic amines. So category amines is the dopamine, noradrenaline or adrenaline, or norepinephrine, epinephrine. Indoamine as well. That's in the serotonin. That's also called the happy neurotransmitters. We will talk about that. So after finish category amine, then we'll talk about indoamine as well. Okay. Now, this pathway biosynthesis, using the tyrosine as the precursor. So this is the structure of the tyrosine. So the first step is the tyrosine hydroxylase. That's the enzyme actually is the limiting step. Okay. So you can see that, you know, tyrosine hydroxylase, adding a hydroxyl group over there. And then the dopa decarboxylase. So we move the carboxyl group, that will become dopamine. Okay. Now the dopamine beta hydroxyl hydroxylase, dopamine beta hydroxylase, add the hydroxyl group over here. And then in the terms of the epinephrine, add the methyl group over here, pentoamine and methyltransferase, PNMT. Okay. Now, from here, you can at once see that, you know, they are not this neurotransmitter, just one hydroxyl group. Then they can distinguish between the epinephrine and dopamine. So the receptors actually is very, very specific in recognition of the molecules. Okay. We will have more of those and how those hydroxyl group difference will affect different type of receptors. So again, we'll talk about the synthesis, storage, release, and uptake as well. Okay. So tyrosine is converted to the DOPA. Okay. The DOPA is converted to the dopamine by the aromatic L amino acid decarboxylase. So there's different names. And the dopamine is transported into the vesicle, and then in the norepinephrine neurons, they will convert to norepinephrine by the dopamine beta hydroxylase. Okay. Because the norepinephrine actually can inhibit the tyrosine hydroxylase. So that's the reason that is transported out of the dopamine and norepinephrine into the vesicle. Okay, there are different drugs that are on here. Now, the vesicle also contain the ATP as well. They contain the enzyme and also the chromogranin, actually, so making the neuron have color. The reason behind is not quite clear. Okay. So the synthesis neurotransmitters, there are several enzymes involved in the synthesis, the tyrosine hydroxylase. So if you still remember the receptor signaling, we talk about the tyrosine hydroxylase, how it is controlled by the phosphorylations and activated. So we will talk a little bit about it. So it is the rate limiting step of the pathway. So it's regulation including the calcium. If you remember that slide, actually with the calcium induced phosphorylations in response to the cyclic AMP. Okay. So the regulations that can be in the cytoplasm. So if it's phosphorylated, then the enzyme activity will be higher, and then that actually can at the time that can synthesize more neurotransmitters. Okay, because actually particularly in the catecholamine system, the neurons actually is the area that it innervate actually is pretty big. But very amazing, those neurons actually, even though there are not too many, but their resilience actually can produce, if that degenerate, that actually can still function until that is going to be really end. Okay. So there's also transsynaptic regulations of tyrosine hydroxylase. If the activity actually is getting more, then that can actually regulate the tyrosine. This is in the case of the sympathetic system. If the presynaptic activity is higher, then that can actually regulate the tyrosine hydroxylase transcription and translation in the cell body. Okay, so increase the amount. So you will see that actually the first regulations in the postsynaptic activity. Okay, in which that the tyrosine hydroxylase activity, the release is increased, the enzyme will be phosphorylated and then increase the activity. Now, another more long-term effect actually is the transsynaptic regulation, in which that the transcriptions and translations of the tyrosine hydroxylase increase. So making more enzyme and also making more active. So what's the difference between these two types of regulation? Anyone? Online student? Face-to-face student? So what's the difference between these two types of regulation? So we have the regulations by the terminal, at the terminal. And we also have the regulation at the transsynaptic regulations. So why would the neurons have two types of this kind of regulation? What's the function of it?
If it's affecting the transcription and translation, so body, that's more long-term effect.
Yes.
Where compared to phosphorylation, those can be reversed.
Yes. Another difference is actually the phosphorylation you can act on fast. But this transsynaptic regulation that will take some time. So you can see that actually when you activate the receptors over here, the acetylcholine receptors, there will be also secondary. So it stimulates these neurons, and then that neurons will stimulate that to release the norepinephrine. At the same time, they will also stimulate some of the other receptors that mediate the secondary messengers, and then that can enhance the transcription level. And also that will have a more long-term effect because phosphorylation actually can be reversed. Okay. So you'll see there's a general depolarization in neurons. So this one, it depends. So that's always how you control depolarization not induced by acetylcholine. In this case, that the depolarization is induced by the acetylcholine. This is in the sympathetic system. Okay. But in the other system, certainly depolarizations in the neuron in general will have a effect, a short-term effect and a long-term effect. So this is, I want to get you into this concept is actually just like in the AMPA and the AMD receptors, if there's just a depolarization not strong, then it might change the neurons, how they respond. But if it's very strong, then that will also change. That's the long-term depressions and long-term potentiation effects. This one is actually a little bit different. This is the sympathetic neuron. So if it is always activated, that means that the neurons needs to be more active and then in the control of the future activity. Okay. So here's the regulation by the tyrosine hydroxylase by phosphorylations. So you can see at a different site. Particularly important is the pKa, in which that is activated by cyclic AMP, in which the calcium dependent protein kinase, calkinase 2, actually at the 40 and 19. And the other one, the cyclic AMP dependent protein kinase, or sorry, the serine residue of 40. Okay. It depends on the calcium, and the less important site are the PKC at the 40, site 40, and the ELK at the 31, the extracellular receptor activated protein kinase. So you see that a lot of those are actually through the calcium. So remember that if you still remember the slide in the secondary messengers, the activations of the synapse to release the neurotransmitter is by the influx of the calcium. So calcium can also have the secondary messengers, then that will activate the pKa and also the calcium calmodulin dependent protein kinase. Then that actually will phosphorylate the tyrosine kinase, tyrosine hydroxylase, and then as a secondary messengers, so that the activity of the tyrosine hydroxylase will increase. Okay, so that will be a faster response. So as I mentioned, the phosphorylation on the either serine 19 and 40 increase the activity of the tyrosine hydroxylase. Okay. However, for the site at 8 and 31, that has little effect on the catalytic activity. So that maybe have other functions, but actually it's not well studied. But the 19 and 40 are the one that actually study quite a bit. Okay. So the catecholamine neurotransmitters stop a little bit of the other enzyme that synthesize it, dopa decarboxylase, or also known as the 1-aromatic amino acid decarboxylase, because it can accept a number of substrates. So it is not very specific. So you can see that it's also involved in the step in the serotonin synthesis. The number 3, another enzyme is dopamine beta hydroxylase. That actually is contain the copper and requires oxygen. Okay. The other one, the phenylethylamine and methyl transferase that transfer the methyl group to the epinephrine. So that is required. This one also required the adenosine methionine for the methyl group. So that's this same reaction also applied to the synthesis of catecholamines in the adrenal gland. So adrenal gland also used a similar method to synthesize the adrenaline. So that's the reason why that the adrenal gland actually is more like a ganglia rather than a hormone secreting gland. But actually, when we talk about the adrenal gland, actually divided into cortex and medulla, and then the medulla actually also involved in synthesis another stress hormone as well. That we will talk about it when we talk about mental disorder. Okay. So we talk about the synthesis, storage, and also release. Now, how about inactivations? So the inactivation is by the two mechanism, the reuptake and also enzymatic degradations. Okay. So there are two enzymes are primarily responsible for their inactivation. So this is also very important. This is by reuptake and degradations. So just a refresh. So acetylcholine, how it is being inactivated? Yes, just degradation, so no reuptake, you see. Glutamate. That's reuptake, okay. GABA. Reuptake. Okay. Now in the category amine, so that will be by reuptake and also by enzymatic degradations. Okay, so there are two primary enzymes responsible for the inactivation of the category amine. Now, one thing is that once it's degraded by the enzyme, that cannot be reused anymore, actually. Yeah, good. The online student also got it. So two enzymes are primarily responsible for the inactivations of the catecholamine, the monoamine oxidase, and also the catechol O methyltransferase. Now they are actually widely distributed throughout the body. So why is the case?
From two different pathways to control.
No, why it is distributed throughout the body.
Oh, I see. Is it also present in bloodstream? Like is catecholamine also present in blood, like just like acetylcholine?
So what catecholamine is present in the blood circulation?
If it's circulating in the blood, then it might affect other organs, I suppose.
Yes. So in the body, the sympathetic system and the adrenal gland will secrete the catecholamine. So that's the reason why if it is in the body, if it is not filled with enzyme that metabolize it, then that will be affecting different organs. Okay, so that's one thing that actually is very important.
It's just like acetylcholine, basically.
Yes. Yes. So that's the reason those that you cannot reuptake as the those neurons. Okay. Maybe I will stop here because I don't want to drag for too long. We have covered quite a bit. So any questions? Online student, face-to-face student? You'll talk about these two enzymes next Monday.
About page 10 of this PowerPoint, the existence of two sites to modulate the activity of TH, is it implies that there are more rooms for the body to modulate the signal?
What signal?
I mean, the release of TH. Is it an advantage that there's more room to modulate?
Yes. There's certainly more room to modulate. It depends on what signal. It can be hormone, for example, steroids, or other signaling mechanism. So this is just an example of how the activity of the preganglionic neuron can have a synaptic regulations on the tyrosine hydroxylase activity. Now, because the tyrosine hydroxylase is the rate limiting step in the synthesis of the catecholamine. Okay, so you will see that actually in some of the drugs, actually you can bypass this and then use it as a treatment method. So if you have more or increase the synthesis, then suppose that you can release more. Now, the reason I mentioned about those, like how to inactivate, so inactivation can be the modulations, can be drug target. So for example, if you have drugs that affect the reuptake, then they can have more neurotransmitter at the synapse, then they can enhance the transmissions. So there are a lot of those examples of how to modulate the transmission. So the reason I talk about it, so this is for you to understand, what is how you can modulate the transmission. But reuptake or enhance synthesis or prevent degradation is not a very good way to modulate it. Why? Think about it, okay? So any other questions from the online student?
Is it because if those neurotransmitters remain within the synapse, it will affect the whole synapse if other transmitters is released?
A little bit close, but not.
I thought one synapse only has one neurotransmitter, right?
No.
Oh, right. Then you can have peptide.
No. You cannot. You don't think it in that way. This is one synapse. The neurons have many synapses. Yes.
But then one synapse is one axon and one dendron, right? And add one axon.
No, not necessary.
Not necessary.
Oh no. It's more complicated. Okay, you can think about it. So we will stop here. Okay. I'll see you next week.